Hello, everyone. Thank you for joining this talk. My name is Jee Bang, and I'm the Clinical Director of the Johns Hopkins Huntington Disease Center of Excellence. I'll be providing the background on upcoming research in HD treatments and briefly highlighting some of them. Then my colleague, Dr. Wenzhen Duan, will be talking about her work on CRISPR and other DNA-based therapies in HD. These are my relevant disclosures. So first, I'll briefly review current therapies in clinical trials, and then I'll talk about new therapeutic approaches. So one of the current therapeutic approaches includes a goal of neuroprotection, and that can come in a lot of different, or that can be done in a lot of different ways. So one way to provide neuroprotection is to reduce degeneration and death of neurons, and to enhance neuronal health and function, and to strengthen neuronal connections. And one drug that's in the trials right now is called predopidine, and it's a pill that activates a protein called sigma-1 receptor, which can provide neuroprotection for the cells in the brain. And another current therapeutic approach is lowering the Huntington protein. So Huntington protein in its mutant form is what's responsible mainly for causing symptoms of HD. So lowering the levels of that Huntington protein would be beneficial. And one way to do that is to do a gene therapy with what's called RNA interference. And that's done by having a harmless form of a virus that delivers the drug that inactivates the messenger RNA from making the Huntington protein. And an example of such a drug is AMT-130, which is injected into the brain tissue with a one-time brain surgery. And there's another way to lower the Huntington protein, and this is done through what's called an antisense oligonucleotide. And an ASO is a piece of synthetic DNA that sticks to the Huntington messenger RNA and prevents it from making Huntington protein. An example of such a drug that's about to go into clinical trial is WAVE-003, and that's injected into the spinal fluid, and it selectively lowers the mutant form of the Huntington protein. And some of these drugs will be discussed in further detail in later sessions. So what's upcoming? So one of the drugs that are coming up is called SAGE-718, and this is a drug that's targeted at treating the cognitive symptoms of HD. And it targets or it modulates the activity of something called the NMDA receptor, which is an important protein for learning and memory in neurons or brain cells. And a small preliminary study showed improvement with cognitive testing in patients with early HD. And this is an oral drug, and it will go into a phase 2 study in the near future. Now, what's upcoming in terms of gene therapies? We've talked about ASOs and RNAi briefly. But there are other approaches that are also gene-based that are coming up. Before we address those, I want to provide a brief background on DNA and protein. So proteins are what are responsible for carrying out many different functions in our bodies. And the DNA contains the codes or the instructions for making different proteins. But DNA first is made into RNA, or RNA is like a copy of the DNA from which the protein is made. So in our case, the HD gene is the DNA, and the instructions from the gene gets copied into messenger RNA, and then from that, the Huntington protein is made. And so knowing this, trying to interfere or target different steps in this pathway is what gene-based therapies are trying to do. So just to go a little bit further into this, so again, CAG repeats, that's what's contained in the Huntington gene. And when it's of normal length, so we're looking at the top half of the screen here, that gets made into this top structure, this normal form of the Huntington protein. So that's what happens normally. But then in this red box, in the bottom half of the slide here, there are too many CAGs in that longer box. So that's an abnormally long stretch of CAG repeats. And that leads to the production of an abnormal or expanded form of the Huntington protein. And so this is a cartoon that shows different points where we can target these therapies. So when you look at these red or orangish sections in the DNA and the pre-mRNA and the mRNA, and the protein in the bottom half of the screen, those orange sections are where the abnormal form of the Huntington gene or protein is contained. And then in the lighter orange boxes are where we can potentially, or we are introducing drug targets. So in the upper half of the picture, we talked about the ASOs and RNAi. And next, I'm going to talk about another place where we can target therapies using small molecules. One example of such a drug is called Branaplam, and it's called a splice modulator. It's also an oral drug, and this is a drug that was originally made for treating patients with spinal muscular atrophy, which is another significant neurodegenerative disorder, mainly affecting children. So when they made this drug, it also happened by happy coincidence that this drug acts on the Huntington mRNA. And it does so by, in this picture here, in the bottom half, including this pseudoexon. And this pseudoexon is normally not included when the Huntington protein is made, but this drug Branaplam forces that pseudoexon to be included. And this pseudoexon is like a signal saying, stop making this protein. So by forcing the inclusion of that pseudoexon, this gives the mRNA instruction to stop making that Huntington protein. So it was found to, in mice, it was found to lower Huntington protein. And also those children who were being treated with Branaplam who have SMA, they were also found to have lower amount of Huntington protein. And a phase 2 study is coming soon for this drug as well. Another gene-based approach is in the circle here, is using DNA repair modulators. So let's take a look at another example. But before we do that, I just want to briefly talk about the concept of somatic instability, which in practical terms leads to further CAG repeat expansion. So some of you may know people with the same number of CAG repeats, and yet they have very different ages of onset of their symptoms. So why is that? And a lot of researchers have looked into that question. And it turns out that some genes, which are called genetic modifiers, can influence the age of onset and the rapidity of the disease course beyond the CAG expansion that the individual was born with. And many of these genes turn out to be involved in what's called DNA damage repair, and they influence somatic instability or further CAG expansion in HD. So there's an example of a protein called MSH3, and this is a DNA repair protein. So normally this protein scans our genes and looks for errors and fixes them, which is a good thing and which is what we want, except when this particular protein comes across the CAG repeats in the Huntington gene, it actually ends up causing even more CAG repeats, which is not what we want. So there's a drug, and it's also an antisense oligonucleotide that can target this specific genetic modifier, MSH3. And so the idea is lowering MSH3 can reduce somatic expansion or further CAG repeat expansion. And this could also be given to premanifest patients. And that trial is coming up as well. So this is a summary slide of some of the therapies that I've talked about. And it's a busy slide, but it really shows all the different kinds of drugs that have been and are in development. So I talked about some of the ASOs and RNA interference molecules and small molecules, and all of these are targeting the Huntington mRNA. And next, Dr. Duan will talk about ways to target the Huntington DNA. And so in this diagram, again, it would be this part where she will discuss the drugs in development in greater detail. And these are some of our center members, and thank you for your attention.
Hello, everyone. Thank you for joining us today. So my name is Wenzhen Duan. I am a professor at Johns Hopkins University School of Medicine. I am a colleague of Dr. Jee Bang. Thank her for a very nice introduction and overview of the therapies coming in Huntington's disease. So my next talk will be focused on CRISPR-Cas and other few gene therapy target to DNAs and a few also target to the RNA therapies. My research is mainly focused on developing therapies and biomarkers for Huntington's disease. So as Dr. Bang already introduced, so Huntington disease is caused by this expanded CAG repeat in the exon 1 region of the Huntington genes. So in order to get rid of all the manifestation induced by the mutant Huntington genes, so there are advanced technology now to target the root of the disease. So I will introduce CRISPR-Cas9 mediated, we call non-allele specific therapies, which most Huntington's patients have both mutant allele and normal allele. So this approach will lower or reduce both mutant Huntington levels as well as normal Huntington levels. And I will also introduce CRISPR-Cas9 mediated mutant Huntington allele specific lowering approach, which is only applied to certain populations of Huntington's disease patients. And also I will briefly touch called the Zinc finger protein mediated mutant Huntington lowering approach. This is also target only mutant allele. So what is a CRISPR-Cas9? So this is a new technology was discovered back in 2012 by two female scientists. Both of them shared the 2020 Nobel Prize on Chemistry for their discovery used CRISPR-Cas9 for gene editing. So in the general language, I would like you to understand that the CRISPR-Cas9 is like a genetic engineered cruiser missiles that can disable or repair a target genes. In this case, we want to disable a disease genes. So CRISPR is a specific guiding sequence which can be used for detect the destroy targeted DNA. The Cas9 is more like molecular scissors, which is an enzyme, can use a CRISPR sequence as a guide to recognize and destroy the specific strand of the DNA that is complementary to the CRISPR sequence. How the CRISPR works. So CRISPR, we can engineer in the lab, engineer the Cas9 proteins with a CRISPR called the guiding RNA sequence, which would be complementary to the CRISPR sequence in the gene. So once this complex was engineered, so we can deliver this into the mammalian cells. So this complex can attach to a matching genetic DNA sequence, which can further down to this cutting the sequence where the guiding RNA target cause this loss function of the genes. Gene cannot be transcribed or translated into a functional proteins. Or they can be engineered to program the DNA to repair genes in certain case. So how our lab uses CRISPR-Cas9 to tackle the Huntington's disease. So use this principle I just introduced. We designed the two guiding RNAs flagging this Huntington exon 1 region, which contained these CAG repeats. And then we engineered this one to a harmless viral vector delivery vectors. And then same time, we also engineered the Cas9. So all combined these two Cas9, the CRISPRs, we delivered them to the mouse brain. So the mouse models we used here called the ZQ175, which is engineered to express mutant human Huntington in their bodies. So these mouse models mimic several aspects of the disease. So the mouse model has abnormal behavior such as motor deficits. The mice also develop brain atrophy, shrink of certain brain area as a Huntington's patient. Those mouse brain also have mutant Huntington protein aggregates. So they mimic several aspects of the Huntington's patient. It's a very nice model for us to develop treatment in the preclinical stage. So we deliver this complex CRISPR-Cas9 into the mouse brains, and then we observe them over months of time to look at how this treatment is going to alter the disease progression. So what we say is, first, we have to make sure the CRISPR-Cas delivered into the brain area we want to be delivered. So we use this fluorescent probe engineered into our guiding RNAs. So by looking at the mouse brains under microscope, you can see this big red area that's called the mouse stratum of the brain region, so which is equal to the caudate putamen area in the human Huntington. So those brain regions, most vulnerable brain regions in Huntington's disease. So we want to deliver the treatment in those areas. So we saw very nicely coverage of the delivery, the gene therapy agent in areas. In a very high resolution microscope, we can see these neurons express the guiding RNA and the CRISPR. About a month later, we take out this mouse stratum, look at whether this CRISPR-Cas9 did reduce Huntington levels. So we look at the mutant Huntington levels, they reduced. And then we look at the total Huntington levels, they also reduced. So this is before we look at the other phenotype changes, we make sure the gene therapy agent was delivered to the proper region, have its role reduce the Huntington levels. And then we observe this motor. We look at the motor function as one of our outcome measures to evaluate whether the treatment works in the mouse models. So this is called the balance beam. As mouse can be a really good gymnastic. So after a few trials, trainings, they can pass this rod quickly, go to this dark box, because mice generally like hiding in the dark. However, when the disease mice, the Huntington disease mice, you get the same training, reach to the certain age, they will have difficulty to pass this beam. So take them longer time to pass the beam. So we use this transverse time indicate the motor function. The normal mice, you can see the blue dot here, like 3 months, 6 months, 9 months. So 3 months, all the mice look same. Huntington mice didn't have symptoms. So we call this stage called premanifest stage. And then by the 9 months, you can see the very distinguished between red dot indicate the Huntington's mice, blue dot is normal mice. The normal mice takes shorter time to pass the beam. However, the Huntington mice take them longer to pass the beam. However, when we treat this Huntington with CRISPR-Cas9, you can see the mice almost perform identical to the wild-type mice. So this is really exciting. We are working on more fine analysis, see the long-term effect and the safety of this treatment. However, we need to keep in mind, so this CRISPR-Cas9 is so cool technology, but this is mediated non-allele specific. As I mentioned, most Huntington patient has one normal allele, one mutant alleles. In this case, the both allele was knocking down or was lowered. So the pros for this technology, those can target all HD populations because we designed the guardian eye target to the CAG area, which all the patient has this gene sequence. But the cons is this permanent disruption of the normal Huntington might compromise the normal function of the Huntington. They might affect the adult neuronal functions. So we are really needs to look at the long-term safety and tolerability of this treatment. That's our lab and other labs work on right now. So another very exciting technology comes of this finding of called single nucleotide polymorphism. So this single nucleotide polymorphism existed in certain population of the mutant Huntington genes and Huntington patients. So by this sequence and advanced sequence technology, we are able to identify which patients have this single nucleotide polymorphism. This single nucleotide polymorphism, also brief called SNP. This SNP detection actually opens up very wide doors for what we call allele-selective treatment for Huntington disease. Or you can think about this more like a precision or personalized treatment for Huntington disease, because our CRISPR-Cas9 will be specifically targeted to the SNP, which will only lower in the mutant Huntington levels, not touching or not altering the wild-type Huntington levels. So that's the ideal case for HD treatment. We want to just lower the mutant toxic genes, not the normal genes. So you might have questions, how many patients have these SNPs? So right now, the most common found two SNP, we call SNP1 and SNP2. Around this 200 patient population observation studies, we found that like 72% individuals have either SNP1 or SNP2, and about 61% individuals have both. So this gives a lot of hope for a large population HD therapies. The CRISPR-Cas9 mediated SNP-specific targeted therapy can become a very promising treatment. Actually, one of the lab from University of Massachusetts already showing this is very promising in the mouse models. So they engineered the mice carry these human SNPs in the mouse models. So they use the CRISPR-Cas9 target to these SNP areas. So they are able to achieve allele-specific lowering the mutant Huntington in the mouse brain. So this is a really exciting time for us to further explore this promising approach. So another gene therapy target the DNA, as I briefly mentioned, called the Zinc finger binding protein mediate gene therapies. What is the zinc finger proteins? So zinc finger protein abbreviation also called ZFP is a small protein structure motif with a finger-like kind of appearance structures. So this specificity of the zinc finger binding proteins, they can be engineered to target the desired DNA sequence. In this case, the patient have normal allele, Huntington allele, and mutant allele. So the zinc finger binding can be designed to only bind into the mutant allele. So, and then we can engineer another called the transcriptional repression domain. So this protein called CRAB, this main function of this CRAB is suppress or eliminate the genes from DNA transcribed into the messenger RNA. So by combined CRAB with zinc finger binding proteins, so they were targeted to the mutant Huntington allele and suppress the mutant Huntington gene express. So this is also achieve the mutant allele specific binding or specific lower Huntington levels in the human brains in the future, hopefully. So above, I talk about the 3 different DNA targeting gene therapies developing in Huntington disease. This what I will talk about another CRISPR-mediated gene therapy which target messenger RNA of Huntington instead of DNA. As Dr. Jee Bang already introduced, messenger RNA is between gene code and the functional proteins. But meanwhile, messenger RNA of the mutant Huntington also can have specific structures which can be a toxic, we call toxic structures. So this mutant Huntington itself can be toxic species as well. So in order to get rid of this, we not only eliminate the toxic proteins, we also get rid of toxic messenger RNA species. What this technology called? So this is called CRISPR-Cas13 technology, which was discovered by MIT scientists back in 2018. It's 3 years ago. So they found actually is another enzyme called 13 instead of 9. The 9 families target to cleave or cut DNA. The 13 families cut messenger RNA or cut RNAs in this specific case. So what we did is we designed, but the principle is very similar as CRISPR-Cas9. They also need one called the guiding RNA. Bring this Cas9 to the area or to the messenger RNA we would cut. So what we did is we screened a bunch of different guiding RNAs. We found very fortunately one guiding RNA can uniquely just cut mutant Huntington messenger RNA, not altering the normal Huntington messenger RNAs. So we engineer this guiding RNA with the CRISPR-Cas13D. This is the smallest enzyme in these families. It's easy for us to engineer into one delivery vectors. So we did the similar injected into the mouse brain. So very excitedly, we found with this specific guiding RNA, the Cas13, we are able to achieve knocking down or lower the mutant Huntington specifically in the mouse brain without altering the wild-type Huntington. So those mouse models is heterozygous. They also carry one allele normal Huntington and one allele mutant Huntington. So use this mouse model, we are able to see whether our approach is allele specific or non-allele specific. In this case, we saw this CRISPR-Cas13 with guiding RNA. We saw it's allele specific that only lower the mutant Huntington. Right now, we are working with our collaborators to look at long-term safety, target specificity, and efficacy in the preclinical mouse models of this approach. So as you heard about all these very exciting, innovative gene therapies, but one hurdle comes to the brain disease is our brain has a strong wall we call blood-brain barriers. So in the normal case, this is good. It protects our brain from they can eliminate any toxic or unwanted molecules getting into the brain, so protect our brain. But this blood-brain barriers also created a big hurdle for many drugs get into the brain because they, especially like gene therapy, they are large molecules. They're very hard to pass or penetrate these blood-brain barriers. Fortunately, there are technology called lipid nanoparticles. You may have heard in past couple of years due to the COVID, because both COVID vaccines, the Pfizer's and Moderna's vaccines, actually were delivered by lipid nanoparticles. The powerful of these lipid nanoparticles, they can wrap basically anything. They can wrap DNAs, proteins, RNAs into these lipid small particles, and then we can engineer to alter or modulate the size of the lipid nanoparticles and also engineer the surface, make these lipid nanoparticles more easy to pass the blood-brain barriers. So right now we are working with our bioengineer scientists, try to deliver or develop more feasible lipid nanoparticles, deliver both CRISPR-Cas9 and CRISPR-Cas13 like large molecules into the brain, so we can kind of deal with these blood-brain barrier problems. Another way is through the lipid nanoparticles, we also can deliver those small particles through intranasal. Just like when you get a flu shot, sometimes the kids can get option for the nasal sprays. So we hope for this will be a future delivery of Huntington therapy, just spray into the nozzle. So the beauty of this is in this specific breath structure behind our nose is called olfactory bulb. This area is lacking the traditional blood-brain barriers. So the drug delivered through the nozzle so can get into the brain without past the blood-brain barriers. So those are few areas we are working on to enhance the delivery of different gene therapies to be able to help achieve the result we like to. Overall, I would like to share some take-home message with you from our work and other labs. We introduced CRISPR-Cas9 mediated non-allele selective mutant Huntington-lowering therapy at the preclinical stage. We are able to delay the onset and slow the disease progression in the preclinical trials. Several allele-selective mutant Huntington lower as zinc finger binding proteins, CRISPR-Cas9 target to SNP or CRISPR-Cas13. Those approaches are very promising for continued development. And the lipid nanoparticle is a very promising potential to effectively or non-invasively deliver the gene therapy to the brain. So this is a really exciting time for CRISPR gene editing therapies. Think about in the 2019, we saw the result from the first human clinical studies about the safety and feasibility of the CRISPR-Cas9. But about a year later, we have already seen the CRISPR gene editing tools insert directly into the human body for the first time for another rare genetic disease. So I know many more clinical trial studies are in these peptides coming. Hopefully, for Huntington disease are coming soon in the near future. So thank you all for your participation to fighting the HD. So we can view like from 1993, from identify the gene caused Huntington disease to 2021 now, how much progress we have been made so far and how many promising therapies are coming in the horizon. Thank you all for your attention.
Okay, thank you for your attention during that talk, and I hope you enjoyed the illustrations at the end, and you can always go back and watch them again. So now please go ahead and type in your questions in the purple box, and we'll go through as they come in. And the first question is, do the trial treatments that we shared today actually reverse the disease or just stop it from progressing? So that's an excellent question. Currently in the clinical trials, these drugs are being tested in volunteers that have early stage HD. So we are really moving towards trying these drugs in those who are premanifest, who have not yet developed the symptoms. So at this point, the main goal is to slow down the progression. And that's the key part about these types of drugs, which are called disease-modifying drugs. So that's the difference between disease-modifying drugs and symptomatic treatments. So symptomatic treatments, like the ones you use for chorea and mood symptoms, they improve the symptoms, which is very important, but they don't actually affect the disease process per se. So first goal is to slow down the progression, but ultimately we would like to reverse the disease or just really actually stop it from even starting, which is the key part about really targeting those who have not yet manifested symptoms. And that's a big focus in the upcoming trials. And the next question is about the Unicure study. Is that the one with the brain surgery? That is correct. That's the one where the drug is an RNAi compound, RNA interference. So that's the one that's going to use the harmless virus that's going to carry the drug into the brain tissue. So there'll be injections of this drug into three different spots on either side of the brain, the caudate and the putamen, two spots in the putamen and one spot in the caudate. And because that's where the HD disease process initially starts. So that's the UNICURE study, and you'll be hearing about it more later as well. The next question is, let's see. So Dr. Duan, when is CRISPR coming to human trials in HD?
Okay, that's really good question. That's also the question we keep in our mind. So right now, the result we presented today is really in preclinical mouse model studies. So there is definitely safety and tolerability and the delivery. This is a major target area we are working on. Hopefully, in the near future, this can move to the clinical trial, but right now it's in earlier development stage. Thank you.
And another one for you, Dr. Duan. Which do you think is the most promising? CRISPR, zinc finger protein, ASOs, splice modulators?
Yeah, I think this is really good questions. We hope we know which one is best. I think that it's really dependent on the patient's disease stage. The overall is because some research already show actually mutant Huntington might also alter some developmental abnormalities. So the earlier is always better in terms of the treatment start. So we hope to start any of those treatment earlier enough to really stop the progression, not just slow down the progression. So we right now, we don't know which one is best. Hopefully, all of them will be good enough to modify disease progression as long as we keep developing this treatment.
And I think it may be that we might need more than one modality. It may end up being a combination of therapies. And speaking of earlier treatment, I think, this one's also a good one for you, Dr. Duan. Is it possible to use CRISPR-Cas technology to eliminate mutant Huntington gene in the pre-implantation stage to get rid of mutant Huntington permanently?
Okay, that's a really good question. So right now, there are another technology called pre-implementation diagnosis, because of the Huntington patient, majority is one normal allele and one mutant allele. So if the people who have Huntington disease, they got in vitro fertilization, so that means they fertilized the egg also have 50% chance to be normal or to be inherited disease. So through this already available pre-implantation diagnostic, so we will know which infertilized eggs are normal. So that is already available in the clinical right now. So we hope future this CRISPR-Cas technology also come to the clinic to be able to engineer and make 100% all this IVF would be normal, but that's still in earlier developmental stage, not into the clinical yet. Yeah, but theoretically that's possible to correct these mutations in the earlier stage.
Thank you. And the next question, I'll take a stab at it, and then Dr. Duan, if you have any additional comments, that would be great. It sounds like it could be dangerous to turn off the normal HD gene or to cut it out. How will you know that is safe without potential long-term dangers? And I think that's a really important question without a super clear answer yet. We know that normal HD gene is responsible for a lot of important functions in our body. But what we are not quite certain yet is how much of it can we get rid of and still be okay. So, we are waiting. We need long-term data in humans. We have some data from animals. But Dr. Duan, do you want to add anything to that in the last remaining
Yeah,
minute or so.
that's the key question we try to answer. Yeah, based on all the preclinical data, is a certain percentage knocking down normal Huntington in adult age might be okay for a while, but that same percentage knock down mutant Huntington already showing improvement symptoms. That was the sum of the clinical trial based result to start it. But of course, yes, that's we want to know how much lower normal Huntington is safe for this non-allele specific knocking down approach. Yeah, thank you.
Great, and we're out of time, but thank you
Yeah.
for all those wonderful questions, and thank you for your attention, and enjoy the rest of the day.
Thank you.